This protocol is significant because it concentrates extracellular vesicles, EVs, through a combination of technologies while allowing for separation of virions away from EVs. This method maximizes EV recovery above the current gold standard of ultracentrifugation for multiple downstream analysis and characterization of virus-free EV preps.
This protocol is ideal for the study of EVs in viral infections. This method can be adapted to other virus systems such as HTLV, Ebola, Zika, and more.
I would expect a first-time user of this method to struggle with nanoparticle preparation, so we recommend carefully following our specifications. Some optimization may be necessary depending on the system.
Visual demonstration of this method is important for illustrating the overall workflow and nuances of the protocol, which will reduce time and mistakes for first-time users.
Start by preparing culture supernatant from infected or transfected cells. Culture approximately 10 milliliters of late log cells for 5 days at 37 degrees Celsius and 5% carbon dioxide, making sure that all medium reagents are free of extracellular vesicles. When the culture is ready, pellet the cells by centrifugation at 3000 times G for 5 minutes and discard the pellet. Filter the supernatant with a sterile 0.22 micrometer filter and collect the filtrate in a clean tube. Add an equal volume of PEG precipitation reagent to the filtrate and invert the tube several times to mix. Incubate the mixture at 4 degrees Celsius overnight. After the incubation, centrifuge the mixture.